Good. So I'll talk about uncovering master regulators using single-cell transcriptomics. So today we're going to dive deep into single-cell sequencing and gene regulatory networks. Don't worry, I'll introduce both topics first, after which we'll dig deep into one of my favorite tools for this kind of analysis. It's called Scenic. I will end with two real-life case studies where we applied Scenic to help clients. So first, as promised, single-cell sequencing. As the name implies, you have to first generate your single cells. This is usually done by dissociating your tissue into a solution, and then either pipetting a single cell into a plate or using an advanced microfluidic device to generate oil droplets containing just a single cell. Once you've isolated your single cells, you can start extracting and sequencing their RNA molecules. After that, you can map that to the transcriptome, and then you'll end up with what's called a gene count feature matrix, or count matrix for short. This is a very large table where the columns represent the individual cells and the rows represent the individual genes. And so for each cell, you have the measured amount of mRNA molecules for each gene. As you can imagine, this table is very large. Hence, your data analysis, or one of the key aspects of the data analysis that I want to highlight today, is the dimensionality reduction that takes place. So you have a high dimensional matrix of around, let's say, 26,000 genes. I plotted here 3 for convenience. Any more than 3 would make your head hurt. So how do you collapse, let's say, this matrix into those nice T-SNE and UMAP plots as shown by Matthias or shown in publications? Well, the answer is dimensionality reduction. And this can be done in multiple ways, but most often scientists extract the top 3,000 to 5,000 most variable genes, run a principal component analysis, and based on that reduced dimensional set of, let's say, 30 principal components, that's what they use to create their visualization. I can talk much more, but I'll move to gene regulatory networks in a bit, after I've highlighted that the sequencing and mapping step and the data analysis step, if this sounded very complicated, is also very much within scope for our OP² platform that Matthias highlighted, and it can take care of all these complicated steps for you in just a few clicks. Now, gene regulatory networks are often associated with these very complex hairball-like figures. They have a lot of dots and a lot of arrows. Again, I'm not going to dive too deeply into this topic. I'm just going to highlight 2 main points which are fundamental to any gene regulatory network, and that's how this method, or each method, selects their co-regulated genes. And so each dot, why does it group these dots in the way that it does? And the jargon is called how you select the nodes. Then your method also has to account for the control of each gene set. So how do the genes regulate each other? This is represented by the arrows, or as they're called in the jargon, the edges. So each method has their own way, and the use of prior knowledge is a key aspect. Use or not use of prior knowledge is a key aspect here. Methods can use very small, very limited prior information, for example, just some genomic information, up to fully validated protein-protein interaction databases to create this kind of network. Of course, you have to take into account that the more experimentally validated model you want to start from, the more certain you will be of the interactions or the predictions the model provides. However, it will produce very little novel models or novel interactions, that were not known before. So there is a balance, let's say, and you have to strike between whether you want to find novel interactions or you want to find interactions that are most likely biologically true. In addition, something to take into account as well is that prior knowledge may not be generated for the biological system, so the species, the tissue, or the cell type that you, for your experiment, are looking at. So this is a very important caveat when creating, interpreting, and using these kind of gene regulatory networks. All right, this concludes the introduction. Now let's move to my favorite tool to do gene regulatory network analysis in single-cell sequencing. So the tool is called Scenic. It has 3 main steps, and they address the key points that I highlighted first. So again, how does Scenic collect the co-regulated genes? How does it connect, let's say, the transcription factors to the genes? To do this, it uses a random forest regression model, so a machine learning model, to infer co-regulation between transcription factors and expressed genes. So it takes in your count matrix, so your cells and your genes, and it looks for gene modules, so genes that are co-expressed and are significantly associated with a specific transcription factor. So this first step creates or investigates correlation, co-expression. Second, Scenic adds in the causal aspect of your network. To do this, it uses a rather minimal amount of prior knowledge. It just uses the genomic information of your expressed genes. So it checks whether the genes in your predicted modules share a motif, so a genetic sequence that corresponds to the binding site of the predicted transcription factor. So this is really a sanity check. Can the transcription factor that I think regulates these genes, does it have the actual ability to do so? Is there the potential? Doesn't mean that it's actually doing that, but can it do that? So based on that, your regulons or your groups of genes get pruned, so only genes that effectively have this kind of the correct motif are retained, while others are discarded. So we end up with what Scenic calls a regulon. So that's a group of co-regulated genes that share a transcription factor motif. So that's the causal aspect of it. Then how does it use these regulons? It again creates a regulon activity matrix. So for each regulon, it will check in each cell how active is this regulon. And this activity corresponds to the joint expression of all the genes in those regulons. If you do this for all genes and all regulons, you get, again, a matrix, which is quite similar to the count matrix, the rows being the cells and the regulons, and the regulon activity scores being the rows. So these are the main outputs of your Scenic tool. So this is the regulon activity matrix. For each cell, you get the relative biological activity of your regulon. And you can use that to create summaries per cell group, for example, per cell type or per condition, to see which regulons are most active and which are the master regulators, which are the determinants, let's say, of your cell phenotype. I'll show examples in the case study of this. Now, you may have noticed that this type of matrix is very much reminiscent of your count matrix, only it has much fewer dimensions, only a few hundreds compared to your full transcriptome. So you could also use your regulon matrix or your regulon activity matrix for visualization, downstream analysis, anything you can do on a count matrix. You can also, or you can use your regulon matrix also as dimensionality reduction. In addition, apart from this regulon activity matrix, it also provides you with a de novo predicted regulatory network. De novo, because remember, your co-regulated genes are really created based on the data you put in and nothing else. So it's just based on your expression matrix. So you know it's specific for your animal, for your species, for your cell type. So you know that you are working with a very new, very novel network. And it's very interesting to look, for example, for the effector genes and which genes are mediating the phenotype, and are there any interesting targets or modulators within that set. So to summarize Scenic produces regulons, and de novo predicted sets of co-expressed genes with a shared transcription factor. Now on to the case studies. So the first case that I would like to highlight is a differentiation process where we were looking at the oligodendrocyte differentiation in the brain. This is very relevant because diseases of the brain that includes remyelination are severely hampered by our lack of understanding of how these oligodendrocytes reproduce and differentiate. So here, what we did is we actually leveraged public, already available single-cell RNA repositories. We combined the trajectory analysis with this network inference. So we plotted, or we created in silico life cycle, let's say, of your progenitor oligodendrocyte precursor cell to your fully mature myelinating oligodendrocyte, with then the objective to find what is driving this differentiation. How does it look? So what you have here is a summarized regulon activity matrix. It shows the average activity of a set of regulons, being the rows here, for each cell type or each cell group, here being the columns. And as you can see, we could focus this analysis nicely on intermediate cell types. So when they are starting to differentiate, what is driving this differentiation, what are the key transcription factors that mediate this, and what are the key genes that are responsible, the key proteins, let's say, that are being transcribed here. So we could really focus on the master regulators of differentiating cell types using this Scenic analysis. The second case is where we had a disease process, and the client was very interested in the effects of the compound or a treatment on the disease process. In this case, we were talking about fibroblasts. So in fibrosis, there is really a excessive formation of what's called myofibroblasts or inflammatory fibroblasts. Here, the client had a very large data set available with multiple compounds, and we performed network inference using Scenic on the total fibroblast population. And so it's a bit more complicated here, but you can already appreciate that you can really use this technique to identify regulons that are both specific to each given cell population, or also, for example, things that scale up during the fibrotic process. And you can really use this type of information to find the switches, the master regulons that drive this fibrotic process. In addition, you can do the same just by grouping or averaging across your compounds. And you see there is a very dramatic difference between regulon usage in your disease and in your control condition. And you can also already appreciate that, for example, that adding a compound attenuates this regulon signature, not fully, but okay, this is very clear that there is a quite promising effect of the compound. So your Scenic can also identify disease master regulators as well as focusing on the compound effects. That was my last slide.